Before founding Novaflow, what were the biggest bottlenecks you personally faced with?
There were multiple bottlenecks that we would experience all the time.
Looking back, how did your early academic and research experiences prepare you for entrepreneurship? Welcome to Life Science Insights Podcast. I'm Sandil Karnanidhi, and today we're exploring AI, data, and computational biology. Our guest for today is Aman Agarwal, founder and CEO of Novaflow, a company building an AI data analysis for biology labs backed by Y Combinator. Welcome to the show, Aman.
Thank you.
Okay. Can you start by telling us a bit about your background?
Yeah, so my background was in computational biology. So I did neuroscience and English for my undergrad. And so I regularly combine those two things in my day-to-day life and kind of see myself somewhere in between like technical, a biologist, but also like, someone creative and like a liberal arts person.
Okay, that's a fascinating journey. So, what drew you into computational biology in the first place?
Yeah, so I started my career very intent on going to medical school. But I realized, through undergrad that, that path wasn't really for me. I found coding throughout my life something to be something that I enjoyed, mainly because I grew up in the Silicon Valley. I was surrounded by coding. And so I was
Yes.
always interested in the intersection of technology and biology. And so computational biology kind of seemed like the obvious choice. Once I took a computational neuroscience class, that kind of solidified for me that,
Okay.
this is something that I'm very interested in. And then I suddenly had decided that I didn't want to do medicine anymore. I decided that computational biology was the thing for me. It almost was like an overnight decision. And then
Okay.
I started looking for jobs in computational biology.
Okay. But being in Silicon Valley, you always surrounded by the right people and right mindset of all the technology guys, right?
Yeah, I mean, I definitely grew up around that. I did my undergrad at the University of Washington in Seattle.
Okay.
So, there was definitely a lot of tech presence in Seattle as well, but much less in the Bay Area for sure.
Yes.
And, but I always kind of consider tech to be like in my blood because of my experience living here and growing up here.
Yeah, that's interesting. Looking back, how did your early academic and research experiences prepare you for entrepreneurship?
Yeah, so I would say it was kind of 50-50. One of the ways that they prepared me for entrepreneurship was by making it very clear that I was not interested in academia.
Okay.
I knew from kind of an early on that I didn't want to remain in academia forever, though I have a
Okay.
really deep respect for academics, and it's something that like I wish I had the patience for.
Hmm.
Um, but mainly I was really drawn to industry because of the fact that I felt like doing technology, you could make a much more bigger impact. Specifically, like what my courses taught me, I mean, in all of my undergraduate jobs and undergraduate labs, as well as my career afterwards, I was very independent in my work. So I worked in a lab where a lot of my job was helping to develop new computational techniques for analyzing long read sequencing data. And so my project was very independent, even as an undergrad. After I left and I joined a lab in New York, I was routinely working on multiple projects at the same time where I was the only computational person. So guiding the projects was very much up to me. And so because of that, I kind of always had an independent mindset towards my work
Mindset.
style, and I was able to like translate a lot of those skills towards entrepreneurship as well.
Yeah, that's interesting. Was there any specific project or experiment where the data backlog problem became impossible to ignore?
Yeah, I mean, I feel like sequencing in general, like
Yeah.
anyone that does sequencing is very aware that the amount of data, the data that you can generate is endless. And because of that, analyzing it can become quite a monumental task. Specifically, when I was working with long read sequencing data, we were generating hundreds of samples of long read sequencing data, which would amount to tens, if not hundreds of terabytes of just raw data that needed to get analyzed. And
Okay.
so in those cases, I was like, oh wow, we have no tools to do anything with this. Like none of our existing pipelines are robust enough to really handle all of this data. And it's a really big computational challenge just to like integrate this data and to start visualizing it, much less actually produce like relevant analysis and like actual findings from it.
Okay, interesting. So is it a single cell or multi-celled?
So this is what we describe as single fiber. So it's kind of in between. Long read sequencing is separate from short read sequencing.
Okay,
Most single cell platforms that people are familiar with use short read sequencing technologies with barcodes for individual cells, typically through like the 10x system.
okay,
This is specifically with PacBio sequencing and Oxford nanopore sequencing. So we get single fiber resolution where each of the fibers at a single specific genomic locus represent different cells, but we
okay.
haven't actually connected the fibers between the different cells. So, it's kind of an in-between of um,
In
single
between single
cell
cell
and
and
bulk.
both. Okay, got it. So before founding Novaflow, what were the biggest bottlenecks you personally faced with?
It's hard to quantify.
Hmm.
There were multiple bottlenecks that we would experience all the time. One was the amount of data. There was never enough there was always too much data than what could be analyzed by the existing pipelines. Another one was the implementation of new technologies. So anytime that we wanted to basically do a new type of analysis or implement a new pipeline, there was a long turnaround time to when from when we actually like got the data back to when we were ready to analyze it. A third was availability of analysts. So despite the fact that my lab, was well funded and, we had the ability to hire, there was still a lack of analysts as compared to the number of people in doing wet lab experiments. This is just a constant problem that exists in, like I would say, 90% of the labs that I've met, where even if you have someone doing analysis, the amount of work you're producing in the wet lab most of the time will outpace this.
Okay, but your analysis involves only the biology data, right?
Yes, mostly biology data, but different types of data from different types of biological experiments can be
Okay,
very challenging to integrate. So, for example, if you have sequencing data, plus you have proteomics data, and you have metabolomics data, you have to basically find a way to integrate all of those together in a way that makes sense for your experiment, which
okay,
might be different for someone else's experiment. Another thing is like, let's say you've been doing one type of sequencing technique for your entire lab, and now you want to bring in a new type of technique. Even though the pipelines are similar, they're not exactly the same. So you'll have to put in a lot of effort just to basically make a new pipeline to first analyze the data and then actually perform the analysis, all of which will take weeks, if not months, to do. And then on top of that, you're limited by the amount of computational resources you actually have. So you can only do so much analysis at once because of how much compute is available to you. Most universities don't have access to like large HPCs, and if they do, they're hard to use. So there really are like I would say five major bottlenecks that it comes when biologists are looking to do analysis. And just to summarize, those five would be the availability of analysts, the size of the data, the time it takes to create techniques and implement techniques to analyze the data, the
okay.
cost of analyzing the data, and the difficulties in reproducibility. So those are all different reasons why like the
Right.
data analysis process takes a really, really long time.
Yeah, and moreover, the HPC is very expensive, right? So it's getting expensive day by day with all this AI and all the GPU and all the stuff, right?
Mhm.
Okay, got it. So Novaflow is building a AI data analyst for biology labs. How exactly does the platform work, and what makes it different from existing lab analytics tool?
So we automate the entire data analysis process for you. So we'll take raw data, analyze it based on the experiment that a scientist has run, and analyze it with the correct pipeline, generate the processed files, and then run an analysis agent that can take that process data and visualize it based on what the user is looking for. So scientists can essentially talk to their data and talk to the platform to understand what's happening in their data, to generate visualizations on the fly, to focus on like specific genes or to focus on specific aspects of their data that they're interested in for their experiment. In terms of what makes it different, there's a few things. One, our implementation of AI is very different from what people are typically thinking about when they think about AI for biology. We're
Okay,
not trying to essentially do full predictive biology where we take your experiment results and then we predict the analysis for it. Instead, we're working alongside the scientists to help them do their analysis more quickly and provide them the tools in order to actually make um, informed conclusions from their science more quickly
Okay.
than they were in the past.
Okay. But how do you handle the computational part in the back end?
So we process the data. Yeah, we'll process all the data from raw all the way to the analyzed version. So, for example, for RNA-seq, if you
Hmm.
have a FASTQ file, we'll take your FASTQ files and then give you the gene expression CSVs. And
Okay.
then from those gene expression CSVs and other supplementary QC files and things like that,
Okay.
run and create visualizations for your experiment. In terms of specifically how the analysis is done, I
Hmm.
can't discuss that.
Okay, got it. That's fine.
We use published and validated pipelines though to run the processing, and then the analysis is done based on the user's intentions.
Okay, got it. Makes sense. AI can be applied to biology in many ways. So why focus on streamlining data analysis as a first major problem to solve?
Because for me, that's where I see the largest problem and the largest bottlenecks and the biggest opportunity for AI to make an impact. AI is really, really good at, taking dynamic information, coming up with a plan and executing that plan in order based on the user specifications, which is essentially what we're doing in science. And if we can tune that to do it in a way that scientists like and are able to direct, we can essentially enable them to do analysis much, much faster.
Okay. AI is there everywhere, right? How do you see the AI growth in life science sector?
AI, I expect to grow very rapidly. There's already a lot of companies that are trying to apply AI models
Yes.
to do predictive biology. I think a lot of them are potentially doing a great job, though I don't have
Okay.
a lot of details.
Okay.
I strongly believe that the best people doing science are scientists. So
Okay.
I want to give them the tools in order for them to be able to do their own science more effectively and more
Okay.
efficiently. And so that's why Novaflow was created.
Yes. Uh, could you walk us through a real world example where Novaflow dramatically reduced data backlog in a lab settings?
Yeah, so in the case of long read sequencing data, we have users that essentially have hundreds of samples of long read sequencing data that normally it would take a few hours to analyze each one, that they have to do it manually. The maximum they could pretty much do at once would potentially be 16 samples, but when you have 380 samples, that becomes very challenging.
Okay.
Instead, with Novaflow, they can directly connect their HPC to our platform. We directly stream the files in, and so instead of taking the normal week it would have taken to just process and analyze that data, for us, it took under 4 hours.
Oh, yeah, that's a drastic reduction of a timeline, right?
Mhm.
Yeah. Uh, what role do you see AI playing in accelerating biological discovery over the next 5 years?
I think AI is going to play a major role. Um, I also think scientists are going to be playing a major role.
Okay.
Biological discovery is riddled with nuance, and to be able to make accurate biological discoveries,
Discoveries, yes.
understanding that nuance is very important.
Hmm.
So there are things that AI is very good at, but capturing all of that nuance is not something that it's able to do yet, but
Right.
maybe we'll be able to do in the future.
Right.
However, we already have scientists using AI all the time, whether it be for like, really easy literature reviews to summarize papers,
Paperwork.
all of that kind of stuff, to find basic lines of code. People are using AI. And so
Hmm.
people are going to continue to use AI so long as it's helping to speed up their work. And so we'll just see that science is going to start progressing more quickly, especially in the academic settings, because they have access to a lot of these AI tools now.
Yeah, that's a nice advancement. Uh, with the rapid pace of AI advancements, how do you balance automation with the need for human interpretation?
Yeah, that's a great question. So, in terms of automation versus interpretation, we believe that anything that can be automated should be automated. What does it mean to be able to be automated, though, right? So, if there is major decision making that's required in like actual interpretation of results, so like, does this normalization method fit? Does this gene fit my phenotype?
Hmm.
Those types of questions oftentimes lean more towards interpretation
Okay.
than they do actual automation. So, a lot of people describe science as both an art and a science, right? So
Yes.
the parts of science that are more art-like, where, researchers are exercising the creative judgment and also looking at data, interpreting it, and making a conclusion or coming up with an inference based on it.
Hmm.
AI is not going to be doing that. AI is not going to automate that. It's going to
Okay.
try, but at
Okay.
the end of the day, it's not going to be, we're still going to need a human in the loop
Human
to
impact.
validate that what it did was
Yes.
correct. Um, on the other hand, like running a FASTQ file and creating a BAM file, that's
Okay.
not something that once it's figured out,
Okay.
you don't have to rewrite the wheel every single time for that, right? So for that kind of stuff, it makes a lot of sense for AI to be automating it.
Right. But still, we need human eyes to interpret the data, right?
Of course.
Yes. Interesting. As a founder, what has been the most challenging aspects of building Novaflow? Is it technology, the team, or the convincing the labs to adopt new tools?
Um, I would say nothing is easy when you're building a startup.
Yeah, you're
Um,
right.
and it's especially challenging when you're building for biology or healthcare
Okay.
for a few reasons. One, the burden of proof and the rigor of your work is much higher. So we have to be extra, extra sure that what we're building works
Okay.
and we're not creating, we're not like creating results that scientists might not be able to use.
Hmm.
The other thing is scientists aren't really in the same types of channels as sales for like traditional tech people. So having to do sales, you have to come up with many more creative ways to do it as opposed to like your traditional B2B SaaS companies. Um, on the flip side, though, it's been amazing because this is a market that's typically underserved. Nobody's building for academics, nobody's building for scientists. People find it to be too much of a technical challenge, and too challenging of a market to sell to really take the leap in order to build in that space. We have companies, in that space, but they typically end up becoming very niche, or they only end up settling to biotech companies due to the larger contract sizes.
Okay.
So we wanted to be one of the main companies that really focused on serving academics and helping them achieve their research goals faster. Of course, we also work with biotech, but
Okay.
helping academics is really like one of the main driving missions of our company.
Okay. So you Novaflow focus only on the academic research, right?
No, we do both. We
Okay.
help academics and biotech companies, but
Okay.
the inception of the company was really focused or really based on the experiences I had in academia.
Academy. Okay. Okay. Got it. So let's have a few discussion on the Y Combinator. So Novaflow is backed by Y Combinator. That's a major milestone for any startup. What was the most transformative part of that experience for you and the team?
So I'm still NYC right now. We're actively in the batch.
Okay.
Um, I would say everybody should do YC if they can.
Okay.
Um, it's an incredibly transformative experience.
Okay.
They teach you the 101 of how to do a startup and
Wow.
how to sell.
Interesting.
YC's core belief is that it's much easier to teach builders to sell than it is to teach sellers to build. Um, and so they operate on that very effectively, and that you can tell that you can kind of see that evidence in the people that they admit.
Hmm.
If
Okay.
Y Combinator is your goal, just focus on building a good company. That's all they want.
Y Combinator is known for helping startups scale fast. How did their mentorship and network help Novaflow journey in the life science space?
Yeah, so YC typically has the notion of that they're primarily focused on B2B SaaS.
Okay.
Um, that's partially true. Most of the companies in YC are B2B SaaS companies.
Okay.
We didn't have a lot. Actually, we had no companies in our batch that were potential customers for us. We didn't have many companies from previous batches as well that were potential customers for us. I think it was less than 10.
Hmm.
Um, However, we do have a very strong network, and the community at YC is very, very helpful. So everyone
Okay.
that's in our batch
Hmm.
that we've met has been very supportive and is very
Okay.
willing to make intros, and help us find people that they know in their network. Um, I wouldn't say specifically for the growth of the company in
Okay.
terms of like finding customers,
Okay.
YC was like instrumental. But I
Instrumental.
will say in terms of the mentality of how to sell and how to reach customers,
Okay.
it's pretty universal regardless of what market you're in, and they really have a lot of really good tips and strategies for that.
Okay. Yeah, that's very important, right? How they teach you to get the customer, right?
I will say though, you can learn everything that you learn in YC just by reading Paul Graham's essays.
Okay. Okay.
Most of the content that we talk about in YC and how to do it is found there. I just was not familiar with it before coming to YC,
Okay.
so for a lot of this stuff, it's new for me.
Okay. Interesting. What's one piece of advice or insight you took from YC that you still apply for your decision making today?
Yeah, I think the main thing is like I just think about YC's core value and it sounds super cliche, but it's build something people want. And I just think about that all the time, every
Okay.
day. Every time I'm making a decision, I'm like, am I doing something that brings me closer to this goal, that I'm building something that people
Okay.
actually want and not just something for the sake of building it?
Okay. Okay. Interesting. Um, how has your background as a computational biologist shaped your approach to leadership and product development?
I think one of the main things is to be a holistic thinker.
Yes. Yes.
um, to really try to get as many of the facts as I possibly can
Mm.
before making decisions,
Okay.
while keeping in mind that decisions have a time limit. Like you can't like you have to do things quickly and you have to keep moving. Like
Okay.
you can't continuously wait around for having all the information, but knowing when you have enough information to make an informed decision is really important. I like to think about the analogy of like, oh, you have cells that are going to die unless you decide what experiment to put them in. But if you wait too long to decide what experiment, then they'll just die and then you can't use them for anything, right? Obviously, that's not how things actually go in the lab, but it's just something that I think about because
Okay.
it prevents me from having decision paralysis.
Okay.
And especially when I feel like, I feel like, I don't have all the information, but I still don't feel comfortable with the decision, but I have to make one. And so
Yes.
that really helps me in those cases because there's no way to know that you
No way.
actually have all the information. You
Okay.
just have to make the decision the best you can.
Yes. Yes. That's true. Uh, what's next for Novaflow? Are there any plans to expand into other areas of lab automation beyond data analysis?
In terms of what's next, we're definitely planning to expand past biology into other verticals for data analysis, like chemistry, physics, other types of research. We want to kind of be the one-stop shop for research data analysis across all fields, not just
Mm.
biology. Um, that's the main thing that we're focusing on. My personal goal would be to also do science, since I still consider myself
Yeah.
to be a scientist.
Scientist.
So,
Okay.
we do want to spend some time and money investing in actually producing science as well, not
Okay.
just enabling others to do it.
Okay.
But that's definitely down the road. Right now, there's plenty of people to help and plenty of market to capture, essentially, of people who need help doing analysis and need tools for doing analysis.
Yeah. Yeah. Sure. A very good roadmap. If you could change one thing about the way biology research is currently done, what would it be?
Limitations on sharing data.
Right. Yeah. That's nice.
Yeah,
Correct.
I think that's the biggest thing that's
Yeah.
very challenging. It's hard
Yes.
for researchers to use other people's data.
And
Um,
and sharing, right?
and sharing, and also once data is publicly accessible, it's still not the
Mm.
most easy to access, which I can understand there are bad actors, but
Mm.
um for technological development to happen and for advancement to happen, people need access to data. And if people can't use each other's data, it just slows down the progression of science for everybody.
And more importantly, computation also, right? The expensive and with all this computation taking a long time, right?
Mhm.
Yeah. Okay. Uh, that's all I have for today. Thanks for joining us today. Hope you enjoyed the conversation. It was truly interesting and insightful. And thanks again for joining this call.
Thank you.